all the steps are not completed. So in plant breeding process, there are 5 main steps. Those 5 steps are not completed in the previous class. Once again, I repeat this step. The main steps in the plant breeding, that is collection of variability, evaluation and selection of parents, cross hybridization among the selected parents, and selection and testing of superior recombinants, testing, releasing and commercialization of new cultivators. These are the 5 main steps which are present in the plant breeding process. So children, this steps of the plant breeding, it is very, very important for essay type question. Okay. First step is collection of variability. Genetic variability is the root of any breeding program. So genetic variability means genetic constituency of the parents are known. First, we have to know about the genetic constituency of the parents. Then after, we have to choose the parents if the parents are present in homozygous condition or heterozygous condition. The parents are always present in homozygous pure line condition. Those parents are chosen for cross hybridization. Generally, pre-existing genetic variability is available from wild relatives of crop plants. Wild varieties are present in the nature. Those wild varieties having number of desirable genes. So that genetic variability is available from these wild varieties. Then the entire collection of plant seeds having all the diverse alleles for all genes in a given crop is called as germplasm collection. Germplasm collection means the specific plant along with its seeds and diverse alleles along with desirable genes are collected. That collection is called as germplasm collection. And coming to the second step, that is evaluation and selection of parents. Evaluation and selection of parents. So the germplasm is evaluated. So as to identify the plants with desirable combination of characters. The collected germplasm is evaluated and that germplasm is either it contains a desirable combination of characters or not. It is find out before cross hybridization. Then the selected plants are multiplied and used in the process of hybridization. Selected plants, those plants who are selected as parents before cross hybridization, they have to multiply because they are less in number. So that's why before cross hybridization, those selected parental plants are multiplied and they increase in its number and used. Then after, those parental plants are used in the process of hybridization. Then pure lines are created wherever desirable are possible. Pure lines means homozygosity is maintained for several generations in the selected parental plants and that pure line condition is finally achieved. Those plants are used as parental plants. These are the steps which are present in this evaluation and selection of parents. Parental plants are always present in homozygous pure line condition. Next, third step, cross hybridization among the selected parents. The desired characters have often combined from 2 different plants, that is male parent and female parent. Male parent having one desirable character. For example, high protein quality of one parent, it is considered as male parent, may need to be combined with disease resistancy, which is the another desirable character which is present in the female parent. Two plants, male and female parents are selected, and male parent with high protein quality and female parent with disease resistance capacity. These two plants are crossed with each other. It results in the formation of a hybrid variety with two desirable characters in a single plant. This is possible by cross hybridization with two parents. So two parents, nothing but male and female parents with desirable characters. Both are crossed with each other to produce hybrids. These hybrids are genetically combined the desired characters in single plant. This hybrid plant, it consists with full of desirable characters. Those desirable characters are getting from both parents, male and female parents. Those desirable characters are present in a single hybrid plant. It is the main aim of plant breeding. So when we observe this cross hybridization in this diagram, this one is the male parent and this is the female parent. Female parent is bisexual in nature. So that's why before pollination in this female parent, male reproductive portion is removed, and then stamens are collected from the male parent. They have to put on the stigma of the flower on the female parent to encourage cross pollination. Due to cross pollination, new recombinants are formed with desirable characters. Due to cross hybridization, new recombinants are formed, means genetic material is exchanged in between the two different plants. It results in the formation of new recombinants with desirable characters. So stamens are collected from the male parent, then that stamens are put on the stigma of the female flower on the female parent. It results in the formation of hybrid seed. So it is the hybrid plant with desirable character. The pollen grains from the desirable plant is chosen as male parent. Male parent means stamens which are collected from a plant. That plant is treated as male parent. Male parent means the staminate parent. It having the ability to produce stamens. That parent is called as male parent. And male parents have to be collected and placed on the stigma of the flower of female parent. Female parent means ovulate parent. It consists ovules and it consists female gametophyte. It is considered as female parent. Okay. Pollen grains are stamens collected from the male parent, and that collected pollen grains are kept on the stigma of the flowers of female parent. In this hybridization, cross pollination is necessary. Cross pollination means stamens or pollen grains are collected from a male parent, and those collected stamens or pollen grains are transferred to the female flower of the other plant, not on the same plant. They are transferred to the stigma of the female flower on the other plant. It is called as cross pollination. Due to cross pollination, some variations are formed. Okay. So in this hybridization, cross pollination is necessary. So that's why stamens on the female flowers on the female plant are removed to prevent self pollination. So in self-pollination, there is no recombinant formation. So that's why self-pollination is prevented to encourage the cross-pollination. So that's why stamens on the female flower, because female plants are bisexual in nature. They consist both male and female reproductive portions. So that's why before pollination on female flowers, stamens are removed to prevent self-pollination and support for cross-pollination. So this phenomenon is called as emasculation. Emasculation means removal of anthers and stamens, anthers or stamens from the female flower before pollination. That phenomenon is called as emasculation. So due to emasculation, self-pollination is prevented and cross-pollination is encouraged. Okay? And this emasculation process is always depends on the size of the flower. When the flowers are larger in size, hand emasculation is practiced. When the flowers are smaller in size, hand emasculation is difficulty. So that's why instead of hand emasculation, the forceps or scalpels are used to remove the anthers and stamens on the female flower. Okay, it is about third step, that is cross hybridization among the selected parents. Next, coming to the fourth step, that is selection and testing of superior recombinants. Selection and testing of superior recombinants. So in this step consists of selecting among the progeny of the hybrid. Due to cross hybridization, hybrids are formed with desirable characters. So that hybrids are having either superior characters or not. So that's why among these hybrids, selection have been practiced if they either consist superior qualities or not. Okay, so this step consists of selecting among the progeny of the hybrid. Those plants have the desired character combinations or not. And the selection process is crucial because it is the toughest process in the entire breeding program. First, we have to identify the hybrid which either consists the desirable or superior qualities or not. So that's why this selection process is crucial to the success of the breeding objectives. Okay, children, breeding objectives nothing but breeding, the purpose of the breeding. If that breeding is conducted for the development of disease-resistant varieties or stress-resistant varieties or high nutritionally enriched varieties, it is considered as breeding objectives. Objectives means the main purpose or aim of the breeding. Okay, the selection process is crucial to the success of breeding objectives, and it requires careful scientific evaluation of the progeny. Okay, in this step, scientific evaluation means the hybrid, it consists superior qualities or not. So that is find out with the help of scientific evaluation. So scientific evaluation is conducted with the help of a standard variety of that particular crop. So this breeding process is conducted in wheat variety. For example, when we take the wheat, Triticum aestivum. So in that wheat, male and female plants are crossed with each other, it can result in the formation of hybrid plants. That hybrids are evaluated with the help of a standard variety in that wheat plant. So wheat, it consists of a standard variety. So that standard variety are old existing variety with desirable characters, which is used as a evaluation tool to evaluate the hybrid plant. That hybrid, it is either consists of superior qualities or not. Okay, so that's why this evaluation, scientific evaluation, is the crucial step in the entire breeding program. Next, this step yields plants that are superior to both the parents. In this step, breeder have to find out the hybrid plants. That hybrid plants are superior than their parental plants, because two parents consist of two desirable characters. Those two parents are crossed with each other and they have to form a hybrid plant, and that hybrid plant is always superior than the parental plant. This step is plants that are superior to the both the parents. That hybrid is always superior than their parental plant. Next, these are self-pollinated for several generations. These are nothing but hybrid plants. Selected hybrid plants are self-pollinated for several generations till they reach a state of uniformity, that is nothing but homozygosity. Means pure line conditions are achieved, also achieved in this hybrid plant. Okay, so in this step, this hybrid, selected superior hybrids are self-pollinated for several generations. Selected hybrids are allowed for self-pollination for several generations till they reach a state of uniformity or homozygosity. Once they have to reach homozygous condition, so that the homozygous conditions are that the characters will not segregate in the progeny. Segregation means they have any chance to change in their genetical constraints. So that's why the segregation is prevented by the homozygosity condition. So that homozygosity condition is achieved with the help of self-pollination for several generations in the hybrid plants to maintain uniformity or homozygosity, so that the characters will not segregate in the progeny. Next, last step in the breeding program, that is testing, release, and commercialization of new cultivators. The previous step, selection and testing of superior recombinants are done, and the last step, testing, release, and commercialization of new cultivators. So in this step, the newly selected pure lines or hybrids are evaluated for their yield and some other agronomic traits of quality, disease resistance, etc. So in this step, the newly selected pure lines are evaluated to study their yield, means productivity. Their yield is more or less. Find out this yield of the selected plant. Next, some other Agronomic traits. Agronomic traits means agricultural characters, that is quality of the crop and disease resistance of the crop, etc. This evaluation is done by growing these in research fields. This evaluation is done by growing these plants in research fields. First, the selected hybrid plants are allowed to grow in research fields, not open agriculture fields. So first, the selected plants are allowed to grow in research fields and record their performance under ideal fertilizer application, irrigation, and other crop management practices. So this evaluation is done in research field and record their performance, record the performance of hybrid plants under ideal fertilizer application. So suitable fertilizers are applied for the particular hybrid cultivation and irrigation methods, suitable irrigation methods and some other crop management practices. Then the evaluation in research fields is followed by testing the material in farmer fields. Listen carefully, children. First, the selected material is allowed to grow in the research field. Then after that material is transferred to farmer field. Evaluation in research field is followed by testing the materials in farmer field for at least 3 to 4 growing seasons at several locations in country. Several locations in country. It is called as multi-locational trials. Multi-locational trials means selected testing material is transferred into the farmer field at several locations of our country and up to 3 to 4 growing seasons and record their performance. So in this multi-locational trials, if we have to find out this, that the testing material is suitable to the diverse environmental conditions, because several locations in our country have different atmosphere conditions. So that's why this testing material is suitable to that particular environmental conditions or not before releasing into the market. Okay, so that's why multi-locational trials are conducted at different places in our country. For example, a wheat variety is developed before releasing into the market. That wheat variety is allowed for multi-locational trials in Karnataka state, Rajasthan state, and Himachal Pradesh. Different locations are present in that particular country. So multi-locational trials are conducted in that particular hybrid crop variety and find out that crop is well adjusted to that particular environmental conditions or not, and their yield is increases or decreases, any change in the yield, and it is well adapted to that environmental conditions or not. Those are checked with the help of multi-locational trials or cultivation at several locations in the country. And finally, the material is evaluated in comparison to the best available local crop cultivator as a check or reference cultivator. Finally, the material is evaluated. Material nothing but hybrid variety is evaluated in comparison to the best available local crop cultivator which is local crop, best available local crop cultivator is used as a check or reference cultivator. So after this comparison with local crop cultivator, it having that hybrid variety or testing material having desirable characters than the local crop cultivator. So it is treated as a new hybrid variety, and finally it is released into the market as a new hybrid variety for cultivation purpose. So it is about the last step, testing, release and commercialization of new cultivators. With the help of plant breeding, semi-dwarf varieties are developed in wheat and rice. So with the help of this breeding program, semi-dwarf varieties means those plants, they are not larger, are not tall, are not short, medium-sized plants. Okay, with the help of plant breeding, semi-dwarf varieties are developed in wheat and rice. So by the production of semi-dwarf varieties, so sometimes when we observe this crop seasons, so due to some adverse changes, like when the crop cultivation period is completed, but due to adverse environmental conditions like high wind and high rainfall, if the plants are tall and the plants are totally bent into the mud or in the soil and the total grains are damaged. So that's why instead of tall varieties, semi-dwarf varieties means they are little bit small and their stems are little bit thick. So due to heavy rainfall and heavy wind, these semi-dwarf varieties do not bend and they remain erect and aerial. So that bending is prevented with the help of semi-dwarf varieties. So that's why with the help of plant breeding, semi-dwarf varieties are developed in wheat and rice. So when we observe the rice, so water is very, very essential for rice cultivation. So due to unexpected rainfall, this rice total crop is totally, due to heavy rainfall and heavy wind, tall varieties of rice is totally fall in the water or mud of the soil and the total crop is damaged in the rice. So that's why with the help of plant breeding, semi-dwarf varieties are developed in wheat and rice to prevent the crop damage due to heavy rainfall and wind. Okay. Then semi-dwarf varieties are developed with the help of plant breeding. So that semi-dwarf genes are present in wild varieties. So all the desirable genes in most of the cases, they are present in the wild varieties. So that dwarf genes are isolated from the wild varieties. They are transferred into the cultivated variety. That is nothing but a wild variety is crossed with a cross cultivated variety. Listen carefully. Wild variety with dwarf genes is crossed with a cultivated variety, which results in the formation of a new variety with dwarf genes, because dwarf genes are present in wild variety. So that's why a wild variety is crossed with a cultivated variety to produce semi-dwarf varieties in wheat and rice plants. Norman E. Borlaug was developed semi-dwarf wheat at Mexico. This person, Norman E. Borlaug, was developed semi-dwarf wheat variety at Mexico country. That wheat variety is called as Mexican variety of wheat. That Mexican variety of wheat also introduced in our country at the time of Green Revolution by Swaminathan, M.S. Swaminathan. In 1963, several wheat varieties such as Sonalika and Kalyan Sona, which were high yielding and disease resistant, were introduced all over the wheat growing areas of our India. So in the year 1963, several wheat varieties such as Sonalika and Kalyan Sona. These wheat varieties having high yielding capacity and disease resistant capacity. These varieties are introduced all over the wheat growing areas of India by M.S. Swaminathan. Next, coming to the rice. Semi-dwarf varieties of rice, Jaya and Ratna. Semi-dwarf varieties of rice, those are Jaya and Ratna, were developed in India. So these are the semi-dwarf varieties of wheat and rice. Next, Saccharum barberi, which is sugarcane. Sugarcane, scientific name is Saccharum officinarum, is the scientific name of sugar cane. Okay. Saccharum barberi was crossed with Saccharum officinarum to get a new sugarcane variety with high yield and thick stem and high sugar quality. Listen carefully. This Saccharum officinarum is the normal cultivated sugarcane variety. Okay. Saccharum barberi is the wild variety. Okay. This wild variety of Saccharum barberi was crossed with cultivated variety of Saccharum officinarum to get new sugarcane varieties with high yield, thick stem, because the sugar yielding in the sugarcane depends on the size of the stem. If the stems in the sugarcane having thick, it yields more sugar. So that's why a cross is made between Saccharum barberi with Saccharum officinarum to get new sugarcane varieties with high yield, thick stem and high sugar quantity. Next, coming to the hybrid maize, Zia maize and Jowar. Those are, in common term, they are called as Jonnalu. Okay, and Bajra have been successfully developed in India with the help of plant breeding. So in maize, Jowar, Bajra, number of hybrid varieties are developed in our country with the help of plant breeding. It is about plant breeding, the main steps of plant breeding and applications of plant breeding. Okay, this question is very, very important for essay type or long answer. Okay, this completed. Next, coming to the breeding for disease resistancy. Disease resistancy or breeding for disease resistancy. wide range of fungal, bacterial, and viral pathogens. These all are microorganisms, fungi, bacteria, and viruses. Those are affect the yield of cultivated crop species, especially in tropical climates. Fungal members, bacterial members, and viruses are with pathogens. They can cause severe diseases on economically important crop plants, and it can affect the yield of that cultivated crop species, especially in tropical climates. Crop losses can often be significant, up to 20 to 30%, or sometimes even total crop is collapsed due to these diseases. So, due to any infection, it is either a bacterial disease or fungal disease or viral disease. For example, the most commonly observed fungal disease is red rust disease on wheat. Red rust disease caused by Puccinia graminis critici. It is a fungal member. It belongs to the class Basidiomycotina. Okay, so a wheat crop is totally infected by a Puccinia graminis critici fungal member. So due to that fungal infection, sometimes 20 to 30% of crop is collapsed, or sometimes the total crop is damaged or collapsed. And coming to the bacterial infection. So the most common bacterial infection is paddy blast disease caused by Pseudomonas oryzae. It can cause paddy blast disease. And when observe the viral diseases, the most common viral disease is tobacco mosaic disease caused by TMV virus, tobacco mosaic virus. So, in this manner, these pathogens, bacteria, fungi, and viruses, they have to infect the economically important crop plants, and they have to damage the crop yield. So that's why, to prevent that infestation, number of disease-resistant varieties are developed with the help of breeding program. So, in this situation, breeding and with the help of breeding, development of cultivators, those are resistant to disease and enhance the food production. By the development of disease-resistant cultivators with the help of plant breeding to enhance the food production, to prevent the infestation of the pathogen. This also helps reduce the dependency on the use of fungicides, bactericides, and insecticides. By the development of disease-resistant varieties or disease-resistant cultivators, to reduce the usage or dependency of the fungicides. Fungicides means the chemical substances which are used to prevent the growth of fungal members. Those are called as fungicides. Next, bactericides. Bactericides means the chemicals which are used to prevent the growth of the bacteria. Those are called as bactericides. So, with the help of plant breeding, disease-resistant varieties are developed. Once disease-resistant varieties are developed, so it results in the reduces the dependency on the use of fungicides and bactericides. Resistance of the host plant is the ability to prevent the pathogen from causing disease and is determined by the genetic constituency or genetic constitution of the host plant. This breeding for disease resistance or disease resistant breeding is always depends on the genetic genetic constitution of the host plant. Resistance of the host plant. It is the major ability to prevent the pathogen from causing disease. The resistance or resistant capacity of the host plant is the major ability to prevent the pathogen from causing disease. And this is totally determined by the genetic constitution of the host plant. Before breeding is undertaken, it is important to know about the causative organism and the mode of transmission. Before going to the disease resistance breeding, first breeder means the person who have to done breeding process to develop a specific new variety. That person is called as breeder. That breeder first know about the causative organism. The disease is caused by a bacterial member or a fungal member or a viral member. First, the breeder was find out the causative organism and the mode of transmission. Transmission means how that pathogen is transmitted. That mode of transmission, it is either through air or water or insects or agricultural tools. That pathogen is how it is transmitted, how it is spread on the crop. The breeder have to know about these two things. First one is we have to know about the causative organism. It is either a bacteria or fungal or viral member. And the second tool is we have to know about the mode of transmission, means how it is transmitted, how it is spread on the crop plant. Next, some of the diseases caused by fungi. Some of the diseases caused by fungi are rust, that is brown rust disease on wheat or red rust disease on wheat caused by Puccinia graminis species, and red rot of sugarcane. This red rot disease on sugarcane caused by a fungal member, and late blight of potato. Late blight of potato disease is also caused by Alternaria, and some other bacterial diseases like black rot of crucifers and some other viral diseases like tobacco mosaic disease caused by tobacco mosaic virus, turnip mosaic disease caused by turnip mosaic viruses. So these are the some of the examples for fungal diseases, bacterial diseases, and viral diseases. Methods of breeding for disease resistance. So there are some methods are there for the breeding for disease resistance. Breeding is carried out by conventional breeding techniques described earlier. Means there are 5 major steps in the any breeding program. Breeding for disease resistance, or breeding for high yield, breeding for more nutrient enriched varieties, or breeding for stress resistance. Any type of breeding program, there are 5 basic steps are there discussed in the previous slides. Okay, so breeding is carried out by conventional breeding techniques or by mutation breeding. Okay. The conventional method of breeding for disease resistance is that of hybridization and selection. So in this conventional method of breeding for disease resistance is based on hybridization and selection of superior hybrid varieties. Its steps are essentially identical to those for breeding for any other agronomic characters such as high yield. So in conventional methods, the basic steps are similar. In those steps are similar to high yielding, disease resistance breeding, or stress resistance breeding. The steps are similar. Hybridization is practiced and superior hybrids are selected and multi-locational trials are conducted and they are finally released as a hybrid variety. The various sequential steps are there. Screening germplasm for resistance source. Screening means to find out to know about the germplasm for resistance source. If a cultivator, the ancestors of cultivator, it consists disease resistant genes. That resistant genes are isolated from the ancestors of that cultivated variety and they are transferred into the new cultivated variety. So in this process, the steps are there. Those are screening germplasm for resistance source and hybridization of the selected parents. Means one wild variety with disease resistant genes are crossed with a cultivated variety. Those two are used as parents. Parents means one parent with disease resistant genes and one parent with high yielding genes. Both parents are hybridized or crossed with each other. Then after selection and evaluation of the hybrid, then testing and release of new varieties. These are the basic steps in the conventional breeding program. First one is collection of variability. Next, evaluation of variability. Next, cross hybridization among the selected parents. Next step is selection and evaluation of hybrids. And last step is testing and commercialization of new variety. These are the basic steps in the conventional breeding program. Some crop varieties bred by hybridization some crop varieties bred by hybridization and selection for disease resistant to fungi, bacteria, and viral diseases are released. Those are represented in this box. So first one is crop name and the new variety name and resistant to any other particular disease. So crop, first crop is wheat. The new variety, the new disease-resistant variety is Himagiri. First crop is wheat, Triticum aestivum. So the new variety is, the new disease-resistant variety is Himagiri, which having resistance against leaf rust and stripe rust and hill burnt diseases. So the new variety of wheat is Himagiri, which having resistance against leaf and stripe rust and hill burnt diseases. Okay. Next, coming to the other crop, that is Brassica nigra, that is mustard plant. So, and the new variety is Pusa swarnim, that is currant rye. Rye is the common name of mustard. Okay, avalu. Okay, so Brassica nigra. In that Brassica nigra, new disease-resistant variety is Pusa swarnim, which is resistant against to white rust disease. White rust disease caused by albugo candida. It is one of the type of fungal member, albugo candida, which causes white rust disease on Brassica plant. So the new disease-resistant variety is Pusa swarnim, which have resistance against white rust fungus. Next, cauliflower. So in that cauliflower, disease-resistant varieties are two. They are Pusa shubra and Pusa snowball K1. These two disease-resistant varieties are developed in cauliflower, and these two disease-resistant varieties having resistance against black rot disease caused by fungi and curl blight black rot disease caused by fungi. These two are fungal diseases, and these two varieties, Pusa shubra and Pusa snowball K1, having disease resistance capacity against black rot fungus and curl blight black rot fungus. Next, coming to cowpea. So this cowpea plant, in this cowpea plant, the new disease-resistant variety is Pusa komal variety. So this Pusa komal variety of cow plant having resistance against bacterial blight disease. So this is a bacterial disease. So this Pusa komal cowpea variety having bacterial blight resistance. Next, chili, Capsicum frutescens. Its scientific name, chili, green chili. Its scientific name is Capsicum frutescens. It belongs to the family Solanaceae. And in this chili, Pusa sadabahar is the new disease-resistant variety is developed in chili. That is Pusa sadabahar. Once again, I repeat the new variety name. That is Pusa sadabahar. This Pusa sadabahar new chili variety is having resistance against chili mosaic virus, and some tobacco varieties also developed, those having resistance against tobacco mosaic viral disease and leaf curl disease caused by some fungal members. This conventional breeding often constrained by the availability of limited number of disease-resistant genes. So the major drawback of the conventional breeding is the disease-resistant genes are available in less limited number that are present and identified in various crop varieties or wild varieties. So when we observe the desirable genes, Most of the desirable genes are present in wild varieties. They are not present in the cultivated varieties. All the desirable genes are present in wild varieties. So that desirable disease resistant genes are limited in number. They are present in wild varieties. And instead of conventional breeding, mutational breeding is used to develop number of disease resistant varieties. This conventional breeding, with the help of conventional breeding, less desirable genes are identified and less number of disease resistant varieties are developed. So that's why instead of conventional breeding, mutational breeding is used to develop number of new disease resistant varieties. So mutation is the process by which genetic variations are created through changes in the base sequence within the gene, resulting in the creation of a new character or trait, which is not found in the parental type. So mutation means sudden heritable changes which are take place in an individual. Those are called as mutations. This mutation was first identified by Hugo de Vries in Enothera lamarckiana plant. Okay, so mutation is the process by which genetic variations are created through changes in the base sequence with genes, resulting in the creation of a new character or trait not found in the parental type. Means with the help of mutations, base sequences are changes in the genes. Base sequences means nitrogen bases, adenine, guanine, thymine, and cytosine. So with the help of mutations, those base sequences are changed in the genes, which results in the creation of a new character or trait, which are not found in the parental type. Okay, so this, which is possible with the help of mutational breeding only. Okay, children, time up. I would like to wind up the class. Thank you, all of you.